Okay, folks, let's talk about the classification of various amino acids. The classification of amino acids is dependent on the R group. So there's nonpolar, polar, or charged R groups. The backbone is always going to be the same, right? The N, the alpha carbon, the C terminus. That R group is what distinguishes the amino acids, and we classify them based on the R group. And in this class, we really focus in on those R groups, and we don't talk quite as much about the backbone. Let's start with nonpolar amino acids. There's a variety of nonpolar amino acids. Some are tiny, some are huge. Glycine is the smallest, where the R group is simply hydrogen. There are 9 total nonpolar amino acids. They come in various shapes and sizes. For example, at the very bottom here, we have alanine. That just has the methyl group. We have isoleucine, which has a longer carbon chain, a longer alkyl chain with 4 carbons. And then phenylalanine that has this benzene ring. But what I want you to take a look at, and this is made more clear through the electron density diagrams that you can see here, is that these R groups are non-polar. You can see how this alanine, how the isoleucine, the benzene and phenylalanine, how these are all kind of green in the electron density diagram. This is because they are not polar. So they don't have that red or blue that we would expect to indicate polarity. They are non-polar. The electron density is spread equally through the R group. The next type of R group is a polar R group. So any type of R group where we could draw a dipole. Often the R group has a hydroxyl, an amide, or a thiol. Probably thiol is something you're less familiar with, so it's like alcohol, but with a sulfur. So SH instead of OH. Some of these, as we'll learn soon, can do acid-base chemistry, which is exciting. I love talking about mechanisms, and we'll see that in a couple of months. Cysteine is a bit of a special case for the polar amino acids, as it can do covalent bonding with other cysteines, so that the sulfur makes a direct bond with the sulfur of another cysteine. It's kind of a special case, but we won't talk about that too much in this class. And so on the bottom right, I'm going to draw your attention to the R groups for these amino acids. So let's take a look, folks. We have the R group of serine, which is OH. You can tell in the electron density diagram that this is a polar molecule. Why? Because you have this red area here showing you the stronger electron density. We also have glutamine. Glutamine's R group is here. And glutamine, if I just draw the R group, okay. Now this R group, you can tell, has some electron density, again, around that oxygen, because it's electronegative, and a little bit as well around that nitrogen, since it has a little bit more electronegativity as well. So these are our polar amino acids, where the R groups are polar or partially charged. Finally, we have our charged amino acids. And I want you to pay attention, folks, because they're called charged because they are charged at physiological pH 7.4. Histidine has a pKa closest to physiological pH. So what we'll learn later on in this class is that histidine readily ionizes. What I mean by that is it's really good at both losing its proton and gaining a proton. And the histidine functional group is also sometimes called imidazolium. Don't worry about that too much. On this slide, I have two examples for our R groups. We have the R group of aspartic acid, which I will commonly refer to as aspartate. Its R group has a carboxylic acid functional group, which is why you see a lot of that red electron density, really pulling those electrons there. Lysine, on the other hand, our group is this alkyl chain that ends in a positively charged amine, and you can see from the blue in the diagram also has some more electron density there. So these charged amino acids are charged at physiological pH. Their R group is what we're talking about. The R group is charged. So those are the three classifications for amino acid.